Okay, so good morning everyone. Welcome to my home office, and today we are going to be covering the two topics. So last week's practice and this week's practice, the central nervous system and the peripheral one. So as I wrote to you in the email, I hope everybody found the new workbook for summer semester. It's in VUCA, and we will be looking at it here together as well, so hopefully you have it ready. And the presentation that I will be showing you is in VUCA in the second and third practice available as a PowerPoint. Also for you to be able to ask me questions, we have the chat available, and I will be checking it from time to time, so please don't hesitate to ask me questions. I hopefully will find out soon enough and be able to respond if I know the answer. I don't have a microscope here, you don't have microscopes, so we will be using online stuff, and we will be using the histology guide that I am sure everybody knows pretty well. So the slides will not be exactly the same as what we have in the classroom, but I will try to make it as useful as possible. So let's start. No questions so far, pretty good. Okay, so the central nervous system we will be talking about, and we will be looking at slides from the brain and the spinal cord. This is just a nice introduction picture from the Clarity method. You can watch the YouTube video, which is pretty cool. So this is a mouse brain made transparent, and you actually see all the neurons and the tracts of the white matter going around. So this should make you curious about the brain. So we will be covering a little bit of theory, and then we will be looking at the online slides. So the types of cells that we will see in general in the nervous tissue, both the central nervous system and the peripheral one, will be glial cells and neurons. So let's talk a little bit about them at the beginning. So neurons that we will be seeing, they will have different kind of morphology, but all of them will have soma. So that's the cell body of the neuron. That's where you have most of the cytoplasm and the nucleus. And then they will have dendrites. So these are the usually shorter processes from the soma, and the dendrites are primarily responsible for receiving the signals that the neuron is getting from other neurons. And then you have one axon, which is the longest usually processes, which is forwarding the received signal onwards. So these are the properties that every neuron will have. However, they can look differently, and so these are the basic morphological types of neurons that we can meet in different parts of the nervous system. So they can be divided based on the number of processes, so some stuff coming from the soma. So the most common type that you will meet most of the time are multipolar neurons, meaning they have multiple processes coming from the soma. So this would be the neuron labeled A. Here you have the soma, and you see one long axon and very many shorter dendrites. So from the soma you have multiple processes coming to all sides. That's the most common one. Bipolar neurons, that's the second example. So in this case, you have two bipolar, two long processes coming from the soma of the neuron. One of them will be the axon going towards the synapses, and the other one will be a long dendrite, which will be then branching off in a distance. So bipolar neuron, then you have, so bipolar neurons could be like interneurons, for example, connecting more of the multiple neurons between themselves. Then you have pseudo-unipolar neurons. So these are called pseudo-unipolar because they look like they just have one processes coming from the body here. So it's the C-labeled neuron. So it seems like they just have one, but it branches off really soon, and it still is one axon going to one side and dendrites going in the other direction. The example of where you can find pseudo-unipolar neurons is the spinal ganglion. So we will get to that in the peripheral nervous system, but spinal ganglion is where you can find pseudo-unipolar neurons. And the last one is very special and quite rare. It's the real unipolar neuron. So from this body of the neuron, all of the processes go in one direction, and then they branch off as dendrites and axon. This really just have one processes coming from the body, unipolar neuron, and you can find it in the retina of the eye, for example. So it's pretty special. We are not going to see it today. So these are the types of neurons based on what comes from their body. Then there is a different system how to divide these the neurons into groups, and that's based on their on their function. So, based on their function, we distinguish between Golgi type 1 and Golgi type 2 neurons. We will also be talking about Golgi staining, so it's probably the same guy. And Golgi type 1 neurons look like this. So the upper one would be a nice example of Golgi neuron type 1. These neurons are responsible for long projections. So they are carrying information from one place in the brain or the spinal cord somewhere else. So long projection meaning long axon. The body is quite big compared to Golgi 2. And examples of this that we will see today will be pyramidal cells, so pyramidal neurons in the brain cortex, motoneurons in the spinal cord, medulla spinalis. So motoneurons and pyramidal cells, we will see that they have pretty big bodies, and from their function is to carry a signal a long way. And Golgi type 2 is the opposite. So these are not forming projections to some other region of the central nervous system, but on the contrary, they are just in one place and they interconnect the neurons like this. So they are called interneurons. They sort of modulate the overall output of the projections that will be carried out by Golgi 1. And they have smaller body and the axon is short. So the axon would be one of these processes. It's impossible for us now to say which one, and they are mostly inhibitory. So you have a network of these big guys forming the projections, and then you modulate, usually negatively, so you tune down the output of the whole network. So that's Golgi type 1 and Golgi type 2 neurons. here you can see them in a slightly more modern staining method. So this is fluorescence staining, and on the left you can see, so this is an example of Golgi type 1 neurons from the cerebellum. We will see these guys later on. These are the Purkinje cells which are sitting in the cerebellum, and so these would be the dendrites coming to one side, branching off, and the axon would be going this way. So you see a long axon, a very big body. So this is typical Golgi 1 type multipolar neuron, and these long axons will go into nuclei of the cerebellum. And this would be a nice example of Golgi type 2, so the interneuron. So it's sitting here, quite small, the soma, and processes coming to all sides, and you can see that there are these small green bumps on the processes, on the dendrites. So these are the dendritic spines. We will get to that as well. So that's where the synapses with these interneurons will be happening. So that's Golgi type 1, Golgi type 2. If you think about the Golgi type 1 neuron, which is a projection neuron with long axon traveling very far away, and it needs to be able to work even though it's very far from the cell body. So you need to carry out quite a lot of transport along the axon, and this is carried out by motor proteins, so kinesin and dynein, and these guys will be using energy, so ATP basically, and they will be walking along microtubules. So along the whole axon you will have microtubules with motor proteins walking along them, and here you have speeded up version of fluorescently stained cargo vesicles that are traveling one way and the other along the axon. What could be carried, for example, to the synaptic terminal would be vesicles with neurotransmitters, for example. Retrograde transport back to the body of the neuron would be some remains of the metabolism, some stuff that needs to be degraded, for example. So axonal transport is pretty important, and when you go along the axon, at the end you reach the place which is called the synapse. So here you see the microtubules, and this is the end of the axon. This is the synaptic button. So it sort of looks like a button a little bit. And there are two types of synapses. You have electrical one and chemical one. The typical synapse that you usually think about is the chemical one. That's the B picture, where you have vesicles with neurotransmitter, you have synaptic cleft and receptors for the neurotransmitter, and that's how the signal is spreading from the presynaptic to the postsynaptic cell. There is also this other type of synapse, which is sort of less complicated. So it's basically just the two cells being connected by gap junctions. So whatever is happening in the first neuron, it will spread to the second one, because the ions will go and the signaling molecules will go through the gap junctions. It doesn't, it's really not so tightly regulated. So in this case, in case of chemical synapse, you can imagine that you can regulate how much neurotransmitter is made, how often is it released into the synaptic cleft, how many receptors you have, which type. But in case of chemical synapse, it's really just what happens is spreading. Whatever happens is spreading, not really tightly regulated. However, it's still pretty important in some cases. So that's the synapses based on the type, and then based on their location, we can divide chemical, so the neurotransmitter synapses, into more types again. So based on what is touching what, sort of. So here you have the presynaptic neuron in yellow, and it's reaching the postsynaptic neuron in different places. So the synapses can either be axodendritic. So the axon will be coming and it will be touching the dendrite of the other cell. That's the most common type of the chemical synapse, and where this will be happening, it will be happening on the dendritic spines that you see here again in the green fluorescence. So this is once again the big neuron from cerebellum, the Purkinje cell, and it will be receiving thousands of synaptic information from other cells on its dendrites, on the dendritic spines. And then there are other options. So you can also have the axon terminal close to the body of the postsynaptic neuron. So that's axosomatic type of synapse, and the third type would be axonal. So the axon would actually be touching the axon of the next cell, not the dendrite or the body. So the second and third type would be more sort of modulatory. So this neuron would be receiving many axodendritic inputs here, and it will be processing them and sending onwards some kind of information, and this neuron would be modulating what the blue neuron does. So it really can get pretty complicated, just for you to know. Okay, so let me check if there's any questions. No, everybody's really quiet. I hope you are watching me. So that was for the neurons, and also there are the glial cells, which are very important in many ways. In the central nervous system that we are talking about right now, there are 4 types that we can see there. And the most common type of glial cells will be the astrocytes. So astrocytes are here in this like greenish color, these guys. And astrocytes are responsible really for supporting the neurons and other sort of stuff that we will get to, and they have 2 versions of them, sort of. So either they can be in the white matter. So white matter is where you have the myelinated axons. So the myelinated axons, we will get to that in the peripheral nervous system more. But so in this case, the astrocytes will be going along the fibers of the axons as well, and they will be called fibrillar. There can be also other names. I think there's other terms for this, but anyway, it's not like a key thing. And in the gray matter, so in the gray matter you have the bodies of the neurons, and there you have astrocytes which are called protoplasmatic, and they will be looking like this, not like fibers, not elongated, but sort of like astrocytes like this. So that's the most common type. Then you have oligodendrocytes. And oligodendrocytes are these guys, and they are responsible for the myelinization. So they will be myelinating axons in the central nervous system. And what is different from the peripheral nervous system that we will be covering in a bit, so one oligodendrocyte will be responsible for myelinating multiple axons. So it's not just one axon, but more of them will be myelinated by one oligodendrocyte. Then you have ependymal cells, and ependymal cells are these ones, and they are sort of an epithelium. So epithelium that will be lining the ventricles and the spinal canal. So one of their functions is to produce the cerebrospinal fluid. So ependymal cells, we will see them in spinal cord. And microglia. Microglia are very different from the other glial cells, and they are similar to macrophages. So they are responsible for the immune reaction, for phagocytosis inside the nervous tissue, and they are also different because they come from mesoderm, as opposed to the other types. So astrocytes, oligodendrocyte, and ependymal cells, they all come from neuroectoderm, like the same as the neurons do, and microglia, they come from mesoderm. So also one more thing I want to mention about astrocytes. So astrocytes, like you are going to see everywhere that they are, supporting, providing nutrition for the neurons. They are also not so passive only. They also play a role in the synapses. So this kind of synapse would be happening between the axon terminal and, for example, the dendritic spine, but the astrocyte will also be involved usually. So this would be called tripartite synapse because you have the presynaptic, postsynaptic, and the astrocyte. So three parties actually taking part, and the astrocyte will be also influencing what happens in the synapse. So it can also modulate its function, and astrocytes can even forward their own signals. So these cells seem to be pretty important. What is another important function of the astrocytes is that they form a part of the blood-brain barrier. So if you think about the brain or the spinal cord, you need a lot of energy for all the processes that are happening there, and therefore you need a lot of oxygen and glucose and stuff like that carried in the blood. So there's a lot of blood vessels, a lot of capillaries inside the brain and spinal cord. However, we need to be very, very restrictive about what comes into the brain tissue itself. So the capillaries need to be really tightly closed by tight junctions. So you have a continuous type of capillaries, not the fenestrated ones or the sinusoids, not at all. You will have the most tight capillary type, and then you will have processes of astrocytes covering the capillary as well. So they will be also taking nutrients up from the blood and forming part of the blood-brain barrier. And the pink stuff that you see around here, those are the basement membranes of the astrocyte processes and of the endothelial cells forming the capillary wall. So all of this will be forming the blood-brain barrier. You can see it here on a picture from light microscope with immunohistochemistry staining. So you see astrocytes as the brown cells here, and you see the processes coming from their bodies to the capillary. So this is part of the capillary on longitudinal section, and you see that the astrocytes are reaching to it with their feet, sort of. So that was blood-brain barrier, and so we will be, let's get to the slides. So we will be looking at both the brain and spinal cord, and from the brain we will be looking at the brain cortex and cerebellum specifically, and spinal cord we will have like a cross-section of the whole spinal cord. Both of these will be divided into white and gray matter, and it will be one of the things we want to be able to do after today's practice is to distinguish between white and gray matter in the nervous system, and to be able to distinguish, to find the large types of neurons, for example, the Golgi type 1. And one more thing we need to talk about before we get to the slides is what staining methods are used for staining the nervous system. So, okay, no questions, perfect. So the nervous system, you can use normal hematoxylin eosin. So some of the slides we will see will be stained with hematoxylin eosin, and so that's the one you already know with dark purple nuclei and pink cytoplasm. but there are other special types of staining. So you can have the Golgi staining, which is this one. So it's the same guy as the Golgi type 1 and type 2 neurons, and this staining is like very nice looking, but it takes like 3 weeks to do, and it's not very frequently used nowadays, and it gives you dark black neuronal bodies and processes. So we will take a look at that a little bit, but most common staining for nervous system will be the Nissl stain. The example of the Nissl stain is this one, the purple one. And so Nissl stain is the name of the staining, but you can use different chemicals. So one of the chemicals that's frequently used is Cressyl violet. So it will be purple, violet, purple. And the Nissl stain in general lets you see acidic stuff. So similar to hematoxylin. So you see nuclei and you see rough endoplasmatic reticulum. So the rough endoplasmatic reticulum, so this would be a big neuron. So big pyramidal cell, for example, in the cortex, and you see a nucleus with nucleolus, and you see in the cytoplasm some purple stuff as well. So that's the rough endoplasmatic reticulum, and it's called the Nissl substance. So just so you know this special term. And another staining that we will be talking about is Luxol-Fast blue. So Luxol-Fast blue is a special staining for myelin. So in case the brain will be stained with Luxol-Fast blue, that could be this example, you will see bright blue where you have the white matter. So so that's it, and let's get to the slides. So in your workbook and in the boxes with slides that we don't have right now, you have brain stained with hematoxylin eosin and brain stained with Luxol-Fast blue. So in these two we will be mainly looking at the cortex and at the border between cortex and the white matter. Then you have a cerebellum, hematoxylin eosin, and cerebellum, Cressyl violet. So that's the Nissl stain. So in these two cases we will be looking at the structure of cerebellum and what layers do you have in gray matter, where do you find the white matter, and finally the spinal cord. The spinal cord will be hematoxylin eosin as well. So let's start with the first slide with the brain hematoxylin eosin. So in here, based on the workbook, so if you look into your workbook, you see the brain cortex and two versions, so hematoxylin eosin and Luxol-Fast blue. And what is our goal to see in this slide is to see the gray matter. Inside the gray matter, which will be the brain cortex, we should find the big neurons, the pyramidal cells. So that will be Golgi type 1, big multipolar neurons. Then we should be able to see the white matter, and in the white matter to see the myelinated fibers. Myelinated by oligodendrocytes, of course. So, based on this, we can start. So, the cerebral cortex, which is the gray matter on this slide, we have 6 different layers. So, this is what they would look like on a nice illustration, and this is how they look like in hematoxylin eosin staining. So you should know that there are these 6 layers with their different names. However, on the slides it's very hard to say where one of the layers begins and where it ends. So it's not necessary for you to say, okay, so this is inner granular layer. No, that we will not be requiring that, but you should be able to find the cortex and find the white matter. We will get to that. And inside the cortex, what is important on higher magnifications, you should be able to find the pyramidal cells. So there are two layers where you can find them. So if we go through the cortex, we will for sure find something like this. So on high magnification, the hematoxylin eosin slide, you will see many cells, because in the gray matter you mostly have cell bodies, and there, of course, the dendrites and everything. And then the axons go away and they go to the white matter as myelinated tracts headed somewhere else. So this would be a nice example of the pyramidal cell. So it's named like this because usually it has a triangle shape, but it doesn't always have to be perfect. And you see the nucleus inside the cytoplasm with nucleolus. So when you see in the brain cortex a big cell like this, it's safe to say this is the pyramidal neuron. What you see around, there's many more nuclei, many more cells. So some of those will be glial cells, probably these tiny ones where you only really see the nucleus, and some of them will be smaller neurons, because you don't only have the pyramidal neurons in the cortex, but you also have the interneurons, for example. So the pyramidal cells are Golgi type 1 and they will be forming the long projection somewhere else. But there's a lot of interneurons, Golgi type 2, smaller ones, which will be there interconnecting these cells. You can see it here. So these are the big pyramidal cells, and their axon will be going away from the cortex. And their dendrites will be very branched and sometimes very long, and they will be also reached by interneurons, which will be influencing what happens to the action potential if it gets sent into the axon. So the granular layers, they have a lot of smaller neurons usually, but in the pyramidal layer you also have many smaller neurons and of course many glial cells, and sometimes you can find blood vessels. So unfortunately, histology guide doesn't have hematoxylin eosin stained brain cortex. So for hematoxylin eosin, you just have to either Google it or let's wait for the quarantine to be over and you can take a look, hopefully until the end of the semester on your own. But we will take a look at different staining on the histology guide. And the other staining that you would have in your slide box, but we don't have on histology guide, is the Luxol-Fast blue. So Luxol-Fast blue will be the staining where you can really nicely find the white matter because it will be blue. And the gray matter, so in this picture you have both the Cressil violet stain and the Luxol-Fast blue. So you can see the cells, the cytoplasm with the rough endoplasmatic reticulum and the nuclei. And so here you don't see the blue staining, so this is the gray matter. So this would be the cortex that we just looked at in hematoxylin eosin, and this would be the white matter. So white matter will be where you have axons myelinated by oligodendrocytes, and the myelin in the sheaths will be stained blue in this case. So let's take a look at what the histology guide
Gray matter
does.
So this would be the cortex that we just looked at.
No. Okay. Jesus. Okay, sorry. So I heard myself talk. And so in histology guide you can find two versions for the brain. So you can see the brain stained with the Golgi method, which is not really our focus today, but anyway, it's you can take a look, it's pretty nice. So here you would see nice Golgi type 1 pyramidal cells in the cortex. So this would be the body of the cell, this would be dendrites coming here, and an axon going away into the white matter. So this is the Golgi staining, but that's just, you know, to know what it is. And the second option is the Cressyl Violet, so the Nissl stain. So let's open that one. And here you probably should recognize that this is not a human brain. However, we can take a look anyway. So this is a brain of this is a rat brain. So it could be either rat or mouse, they're really similar. So this is a rat brain on sagittal section. So it would be, you know, cutting through the brain like this. Coronal section would be like that. Sagittal section like this. So this is sagittal section, and here we see the cerebellum, which we will take a look in a minute, and this is all the rest. So the cortex will be these layers surrounding the two hemispheres. So this is one of the hemispheres cut like that, and if we zoom in, we can see that all of these are layers of cell bodies. So this is the brain cortex with the different layers. So we don't have to get into that right now, plus it looks a little bit different in the rat. But here you can nicely see the difference between the gray matter and the white matter here. So this is the white matter, so let's zoom in. And so in hematoxylin eosin the difference is not so big, but in the Nissl stain the white matter is really actually quite white, so that's nice. And when we get really close, you see that in the gray matter you have many neuronal cell bodies, big cells sitting everywhere around. So that's the gray matter, and this part is the white matter. So you see nuclei, and these will be mostly nuclei of the oligodendrocytes and other glial cells. So for example, the fibrillar type of astrocytes. And the axons that we don't see here really will be going in the white matter, in this case from here to there, or like vice versa, in this direction. So this is not ideal, but so sorry about that, but we don't have to really I think get into the cortex very much, but so the difference between gray and white matter is the most important thing here, I guess. So Cressyl Violet stain, you will see cells, their nuclei, and you will see also the rough endoplasmatic reticulum in the cytoplasm stained pretty dark. So this is how the brain cortex in Nissl stain in a rat brain would look. So this is hematoxylin eosin, and what we saw just now was the Nissl stain. So hematoxylin eosin gives us more information, I would say, because you see the whole cytoplasm pretty nicely. But never mind, let's continue to the cerebellum. So to sum up the brain cortex, we saw the gray matter with very many cells. Some of them were very big. We saw that mostly in the presentation, and they were the pyramidal cells. Then we saw the white matter, and in the white matter there are the myelinated fibers. We will see those in the peripheral nervous system as well. Now let's continue with the cerebellum. So cerebellum is this part of the brain, and it is like an older part of the brain, so its cortex doesn't have so many layers, so which is nice for us. So you will have white matter, and then you will have the gray matter, the cortex as well. So this is the whole cortex here, and it will have 3 layers. So the outer one will be molecule layer, then we will have the cell bodies of the Purkinje cells, these huge neurons, the Golgi type 1, and they will be forming axons going away into the white matter. And the inner layer, where you will have many smaller neurons, will be called the granular layer. So molecular layer outside, then one layer of Purkinje cells, and then granular layer here. And on a slide with hematoxylin eosin, it will look like this. So on small magnification, you can see that the cerebellum is very easy to recognize. This slide is, you can see it like you don't even need a microscope to recognize this is cerebellum. And this structure is called the arbor vitae, so like the tree of life. It's branching, so it sort of looks like a tree. And if you zoom in more, you start seeing the different layers more clearly. So this lighter layer will be the stratum moleculare, the molecular layer. So this is the surface of the cortex of the cerebellum, stratum moleculare. That's where you have a lot of dendrites of the Purkinje cells and some interneurons and glial cells. So it's not so many cell bodies, so it's kind of light pink. Then you have a layer of these huge neurons sitting in a row. So those are the Purkinje cells, and they are the major projection neurons of the cerebellum. So they are the cells that will be forming the final output of the cortex. And the very purple dotty layer here is the stratum granulare. So the layer of smaller neurons, granule cell layer, and inside here this would be the white matter. So the white matter is covered with stratum granulare, Purkinje cells, and stratum moleculare. And this will be happening in all of these parts of the cerebellum, which are branching like this. And in the white matter, not so many cells, and there will be the axons of the Purkinje cells going away from the cortex. So if we zoom in hematoxylin eosin, this is what we will see on the border between the lighter layer of the cortex and the dark one, between the molecular and the granule cell layer. And these will be the huge neurons, the biggest neurons that you can see in the brain, Purkinje cells. So they are sitting here in the row. You can see the cytoplasm with the dendrites reaching upwards into the molecular layer, and their axons will be going down through the granule cell layer into the white matter. So you should be able to find and name these layers and find the Purkinje cells on your slides in the future. So let's take a look at the Nissl stain again, because there's no cerebellum slide either. So let's zoom out. We saw it in the presentation with hematoxylin eosin, and this is the cerebellum with crasil violet again. So it's still pretty, and we can zoom in and take a look at the different layers. So even from this far away, you can see that the inside will be the white matter. So the white matter is here. In hematoxylin eosin it was pink, but in Nissl stain it's really quite light, because it doesn't have that many big cell bodies. So white matter, and around it you have a dark layer, so the granular one, and the molecular layer around. And this is the gray matter. So white matter inside, and all of the rest is the gray matter coming around. So let's zoom in. So this is the molecular layer of the cerebellum cortex. Here you see, not so beautifully, but they are there, the Purkinje cells sitting in a row. Very big cells, nucleus, cytoplasm with rough endoplasmatic reticulum. So this is the Purkinje cell layer, and this one, the darkest one, is the granular layer, and this is the white matter. So its cerebellum is really friendly slide. It's really clear, not very hard to distinguish between the layers. So in the white matter you will see the myelinated axons going away. So molecular layer, Purkinje cells, granular layer, and white matter. Pretty easy. In the workbook it says that you should be able to see the gray matter, we saw. Molecular layer, we saw. Purkinje cells we can find. Granular layer, very easy to see. Purkinje cells are the big neurons sitting there, the Golgi type 1 multipolar neurons, and white matter is inside the cerebellum, forming the branches of the arbor vitae, and it contains the myelinated axons going away into the nuclei of the cerebellum. Yes, so first question, perfect. So are pyramidal neurons specific for cerebral cortex? Yes, they are. So there can be big Golgi type 1 neurons in other places like the spinal cord, but in the cerebral cortex they are called pyramidal cells or pyramidal neurons. So they are specific for the cortex. That's how they are called there. In cerebellum, these big neurons are called Purkinje cells. They don't have the pyramidal shape, so probably that's why they have a different name. Principle is similar. So this was the cerebellum and brain cortex here, and now let's move on to the spinal cord. So the spinal cord will also have gray matter and white matter. However, it's important to realize that opposed to the brain, here you will have the gray matter inside, surrounded by the white matter. So it's reversed. In the brain, on the surface you have the cortex. In the cerebellum as well, on the surface you have the cortex, which is gray matter, many cell nuclei, many cell bodies. And in the spinal cord, the white matter is surrounding the gray matter. So it's the other way around, and white matter will be where all the tracts going up to the brain and down to the periphery will be going. So a lot of myelinated fibers here and a lot of oligodendrocytes therefore, and the gray matter is forming this butterfly shaped area in the middle. So that's where we will see the neuronal bodies again. In the middle of the whole thing you have the central canal, and the central canal is where you have the cerebrospinal fluid going, and it's lined with the ependymal cells, so the type of glial cells that I was mentioning at the beginning. Ependymal cells, they form a simple epithelium which is cuboidal to columnar. We will take a look at it, and these cells also have kinocelia, because they also help the flow of the cerebral spinal fluid. And we will be looking at the differences between white and gray matter. We will take a look at the central canal, and we will try to find the motoneurons. So these will be, so in cortex we saw pyramidal cells, in cerebellum we saw Purkinje cells, and in spinal cord we will have cells that will be big multipolar type 1 Golgi neurons, and they will be called motoneurons because they are the cells that are really in control of your muscles, and they will be in the gray matter in this area. So this is what the spinal cord would be looking with fluorescent staining, and the motoneurons are this group of cells, really big neurons sitting in the anterior horns of the gray matter of the spinal cord. This is the central canal. So let's start. So the difference between the gray and white matter will be pretty clear in case of spinal cord. Again, the gray matter will be sort of more homogeneous, more pink, and it will have many cell bodies. However, not every time they will be the huge motoneurons. They are only in one part of the spinal cord. And the white matter will have more white empty spaces. Why is that? Because the myelin that is covering all the axons is very fatty compound. It's something like lipid a little bit. So it's the same situation as we had, for example, with a white adipose tissue. When you prepare the slides, it washes away. So in these holes you had a lot of the myelin around the axons. So this would be the white matter, this would be the gray matter, and we will get to the slide, but we will be looking for a part of the gray matter which will contain huge cells like this in the anterior horn, and they will be the motoneurons. So white matter, a lot of empty spaces, and here homogeneous pinkish gray matter with the big motoneurons. Axons from the Purkinje. Okay, so sorry. So we have one more question going back to the cerebellum. So let's just, before we go to the spinal cord slide, let's get back to that. So Purkinje cells, they have the axons, and the question is, where are they going? So the answer is they are going into the white matter. So Purkinje cells projections. So let's take a look. So these it's very small. Well, Wikipedia, here we go. So the Purkinje cell is sitting here, and it's sending the axon through the granular layer where you have the small granule cells, and the axon is going all the way into the white matter. And through the white matter, the axon will reach nuclei in the white matter of the cerebellum. So the cerebellum is processing all kinds of sensory information. It's making you to be able to stand straight and coordinate your movements. And to be able to do that, it needs really complex processing. So that's what happens in all these regions of the cortex. And then the cells that are sending out the complex information are the Purkinje cells, and the information is carried through the white matter into nuclei, so groups of other neurons inside the white matter of the cerebellum, and from there to other parts of the central nervous system. So I hope I answered that question. And the dendrites of the Purkinje cells, they are filling basically the molecular layer. So that was going back to the cerebellum, and now let's continue with the spinal cord. So the histology guide has spinal cord with hematoxylin eosin, so perfect for us. Let's open it, and this is it. So this is actually really similar to what you would have during the practices, and so this is a cross section of the whole spinal cord. So the spinal cord would be going like this, and it would cut across. And here you see the butterfly-shaped gray matter inside, and all of this surrounding it is the white matter. What you have around the nervous tissue are the covering layers of the central nervous system, the meninges. So we are not going to be talking about those, but that's the pia mater, dura mater, and arachnoidea, the three layers covering the brain and the spinal cord. So we want to be able to distinguish between the gray matter and the white matter. So let's zoom in. So this is the border between the gray matter here and white matter here. So you can see straight away that it's pretty different. In the gray matter you see many cell nuclei and cell bodies of the bigger neurons. In the white matter you see a lot of little round shapes, and these are the myelinated axons. Because we cut across the spinal cord, we also cut across all the tracts of the axons going up and down. So the axons are also on cross section. So if we zoom in more, so each one of these round shapes would be where you have the axon and the myelin sheath, which was washed out most of the time. So these are all myelinated axons, and of course there will also be nuclei of oligodendrocytes. So this is white matter and this is gray matter. Gray matter, homogeneous pink, filled with many dendrites, astrocytes, and you see big cells here because we are in the anterior horn. So if you take a look from far away, you can you can right away say which part is the anterior horn with the motoneurons. So it's these two parts. These are the ventral horns, ventral, anterior ventral and posterior dorsal horns here. So we want to zoom in into this part and take a look at the motoneurons. And they are here, pretty nice. So you have cell body, stained rough endoplasmatic reticulum, nucleus with nucleolus. So this is how the huge motoneurons will look like. So these are the cells sitting in the spinal cord that will be responsible for making your muscles move when you want to. So Golgi type 1 multipolar neurons, and they are called motoneurons because they are responsible for your motorics. Okay, so spinal cord, and we also wanted to take a look at the central canal. So in the middle you see the hole, which would be filled with cerebrospinal fluid, and when we zoom in we see a layer of, in this case more cuboidal cells lining the spinal canal. And so these are the nuclei sitting here, ependymal cells lining the canal, and they would have kinocelia, but they are quite torn in this case. So ependymal cells here. Also you can see some blood vessels. So this would be a blood vessel which will for sure be a continuous type of capillary, and it will have processes of astrocytes, but we don't see them because we don't have a staining that would be specific for astrocytes. So you don't have to worry about this, but there are blood vessels here as well. These are all big neurons, and the small nuclei will most probably be glial cells, such as astrocytes and microglia and stuff like that. Okay, so that was spinal cord, and that's the end of the first part. So we got through the central nervous system. So we covered, let's review this, we covered the brain cortex, hematoxylin, eosin, and we know about look so fast blue now. We saw gray matter, pyramidal cells, white matter, and myelinated fibers in cerebellum. We covered all the layers, and in spinal cord we looked at the white matter, gray matter, and the central canal, and we saw the motoneurons inside the anterior horns, the huge multipolar neurons sitting there. So now we can move on to the peripheral nervous system, which will be shorter, I think. So let's start. So now we talked about the brain and the spinal cord. That was the central nervous system, and now we will be talking about all the rest. So the peripheral nerves and the ganglia in the periphery, and part of the peripheral nervous system are also the receptors in the periphery, but we will not be looking at those today. So peripheral nerves are all the axons, groups of axons that are going from the central nervous system and into the central nervous system. So all of these yellow lines, and ganglia are groups of cell bodies in the periphery. So usually the cell bodies are in the central nervous system, and just the processes of the neurons go out into the periphery. However, in some cases, in cases of the autonomic nervous system, you will have also cell bodies of neurons inside the periphery, and they will be called ganglia, the groups of them. So when you have a group of cells in the brain, for example, you have basal ganglia. So that's also groups of cell bodies inside the brain. However, other groups of neuronal bodies that are not in the brain cortex, they are called nuclei. So I already said something about nuclei in the cerebellum, the Purkinje cells reaching nuclei, so groups of neuronal bodies in the white matter of the cerebellum. So in central nervous system, these would be called nuclei, like nucleus accumbens or whatever. And the same thing in the periphery will be called ganglia. So ganglia are groups of cell bodies of neurons in the periphery. The peripheral nerves that we will be looking at, they also can be divided based on which way they carry the information. So some of them will be carrying the information from the skin and your sensory organs, so from your eyes, ears, from different sensory receptors in the skin, pain receptors, everything. So all the sensory informations will be carried into the central nervous system by afferent nerves. So skin, an example here. So when you, for example, touch something really hot, you activate the nociceptors, so the pain receptors in the skin, and the information is very quickly carried into the spinal cord. And so this is the afferent part. And then you need to carry out the response to the burning yourself on the hot stove, and that's where the motoneurons come into play. So motoric afferent nerves will be coming out of the central nervous system to the effector organs. In this case, this will be the muscle. So you will touch the hot surface, information will be carried into the spinal cord. Here it will the neuron will form a synapse with the motoneuron. It will tell it, okay, we burned ourselves, and the motoneuron will carry the information to the muscle, and you will quickly move your hand away. So this is the most straightforward example, is the reflex. So some reflexes are really this fast because we need them to be able to protect ourselves, but usually it's not this simple, and you get sensory information coming in, and then from the spinal cord they go to the brain, then from the brain they go, some other informations can go out and into motoneurons, and so it doesn't have to be just one synapse, but it can be many more. So afferent and efferent nerves. And let's get to the glial cells that are specific for the peripheral nervous system. So in the central nervous system we talked about oligodendrocytes that were myelinating the axons in brain and in spinal cord. In the peripheral nervous system, it will be the Schwann cells. So we will be talking about Schwann cells, but let me just go back to this slide, because not all the fibers, not all the axons have to be myelinated. That's important to realize. In the central nervous system, in the peripheral nervous system, there can be myelinated and non-myelinated fibers. The difference will be in the speed of the traveling of the information along these fibers. So, what will happen during the myelinization process is that the cell plasma membrane will be wrapping around the axon. The reason for this is to isolate the axon, which is where the action potential, the change of the polarity of the membrane along the axon, it's traveling. And you need to carry the signal quite a long way. And if you don't want to lose some of the power of the signal, you need to have sort of something covering the axon which will be separating it from the outside. So something like if you have a wire, you usually have the wire covered in a rubber layer, which is doing basically the same thing. So the isolation layer. And in the peripheral nervous system, it's the Schwann cells that are responsible for this. So Schwann cells are once again glial cells which have myelin in their plasma membrane. So both oligodendrocytes and Schwann cells will have plasma membrane which will have special glycoprotein. So glycoprotein tells you it's a protein with a lot of sugar residues hung on it, and it's in the plasma membrane. So the plasma membrane will be wrapping many times around the axon, and in the end it will form the myelin sheath. And this enables the so-called saltatory conduction of the signal, the action potential. So saltatory means like jumpy. So the action potential, the change of the voltage on the membrane will jump from one place to the other, and in between them you will have the myelin sheath, which will not enable spreading of the signal unspecifically and dissipating the power of the signal into the surrounding area. The result of this will be that the speed, for example, of the motoneuron signals will be up to 120 meters per second. So it's very fast. That's the speed that will be happening where you have the reflex, for example, like I mentioned before. And the areas where you do not have the myelin sheath are called the nodes of Ranvier, and that's where you will actually have the ion channels on the membrane of the axon. I will go back to the electronogram, but this is a nice picture to talk about this. So the axon, the depolarization, so the change of the membrane potential is spreading along the axon. That's the information that is being carried from one neuron to the synapse with the other one. And here you have the myelin sheath, which is enabling the saltatory conduction. And in the nodes of Ranvier, you have the depolarization really happening. And then it spreads to the next node of Ranvier and to the next one. If there is a damage done to the myelin sheath, what happens is the current, so the action potential is leaking from the axon to the sides. So it's the strength of the signal is being reduced and the spreading becomes more slow. However, the damage is usually reversible, so the Schwann cells are able to remyelinize the axon. However, it takes quite a long time. And this is a picture from electron microscope where you see a myelinated axon on a cross section. So this is the myelin sheath, the many layers of the plasma membrane and cytoplasm of the Schwann cell going around and around, forming the sheath. And this is the cytoplasm inside the axon, where you see these tiny structures. So these would be the microtubules. So we talked at the beginning about axon transport. So along the axon, which we cut through like this, you have microtubules going, and on these microtubules you have the vesicles traveling on motor proteins. So these are the microtubules on cross section. These would be some of the cargo vesicles, probably. This could be a mitochondria. This I'm not really sure. So this would be a myelinated axon. However, there are also the non-myelinated fibers, and these axons are not totally unprotected because they are engulfed in the cytoplasm of the Schwann cells. So here we don't see the wrapping. We just see one layer of cytoplasm and plasma membrane around the axon anyway. And because we don't have the saltatory conduction, we have sort of the normal, the continuous one, and this leads to a much lower speed. So it can be even under 1 meter per second. So up to 120 in the myelinated fibers, and here even below 1 meter per second. And the non-myelinated fibers, they therefore will be carrying information that's not necessarily transmitted so quickly. So for example, the autonomic nervous system, parasympathetic, sympathetic nervous system will be not required to be so speedy, so it will use non-myelinated fibers. Or for example, there are some examples of pain that are transmitted like this. It's called the slow pain, and it's the pain that comes from your internal organs. So it's not like a pinching pain, but it's sort of more vague feeling, and it's also transmitted quite slowly. And this is what the non-myelinated axons will be looking on a cross section under the electron microscope. So this is the nucleus of the Schwann cell, and its cytoplasm is gray here, and it's surrounding these axons. So non-myelinated axons looking like this. And now we will go to the slides. So no questions, perfect. So we have 2 slides only for the peripheral nervous system. We have autonomic ganglion and we have peripheral nerve. So I would start with the peripheral nerve. It seems more logical to me, and then we will get to the ganglion. So the peripheral nerve, what it really is a big group, a bundle of nerve fibers. And this whole bundle is covered in connective tissue, and this whole thing is what we call a nerve. So it's not just one axon, it's many, like hundreds of them. And so in one of these peripheral nerves, you have both afferent and efferent fibers. It's interesting to realize that one nerve that you see in the body is not just carrying information one way, but it's usually both ways at the same time. And you have different layers of the connective tissue which are protecting and filling this whole thing. So you have endoneurium, perineurium, and epineurium. So these 3 layers are present in the peripheral nerve. So if you have the peripheral nerve on a cross section, so it could look something like this. So you have a group of bundles of axons, and inside these bundles you have the individual axons that we will take a look at, and between them you have the endoneurium. Then you have these bundles covered in the perineurium, so again connective tissue around, and the whole peripheral nerve is covered in the epineurium, so the outermost layer of connective tissue. So when we look at it up close, the endoneurium will be between these individual axons, so the pink stuff, and it will be loose connective tissue, so nothing really special. So you should know that there is something called endoneurium, that is loose connective tissue, and where to find it. We will take a look at the slide in a minute. Perineurium will be dense connective tissue surrounding the bundles of the axons with the endoneurium inside. So perineurium and epineurium will be the cover of the whole peripheral nerves. So collagen connective tissue again, and it's protecting the nerve. So in places where you need extra protection, like in joints, for example, when you don't want, the nerve to get damaged, you will have a thicker epineurium. So let's take a look at the slide. We will use the histology guide again, so you can find somewhere, yes, here you can find the slide of the peripheral nerve in hematoxylin eosine staining, and you have cross section here and a longitudinal section here. So we will take a look at the cross section. That's where we can really see the different layers of the connective tissue. So this whole thing will be one peripheral nerve. So you can see straight away that you have a lot of connective tissue going around the whole structure. So that will be the epineurium. So epineurium will be this layer of connective tissue going around. And all of this inside is bundles of axons cut across. So let's zoom in somewhere, maybe here. Yes. So all of these are the myelinated axons on cross section. So myelinated fibers on cross section, and between them you have a little bit of loose connective tissue. It's really tightly packed, so it's not easy to see, but maybe here a little bit. In the loose connective tissue you would see fibroblasts, and you would also see blood vessels. So this would be a capillary here, and all of these are the myelinated axons. Also there are bigger cell nuclei, and these would be nuclei of the Schwann cells. So think about the axon and it being wrapped in the cytoplasm and membrane of the Schwann cell. So this could be a nucleus of the Schwann cell, and it's wrapping its cytoplasm around this axon. So endoneurium will be between the axons, this pink stuff between the axons. And then when you zoom out, you see that you have individual parts of the nerve. So groups of axons that are sort of separated. And they are separated by connective tissue again. So this will be the perineurium. So endoneurium in here, perineurium around the bundle of the axons. So this would be the perineurium going around. So more dense connective tissue with blood vessels, fibroblasts. See the elongated nuclei of the fibroblasts, and collagen fibers going around the groups of axons. So this is pretty easy. if you get the slide, you really need to zoom in all the way to see the axons. So otherwise it's really hard to say what you're looking at. And but in some areas it doesn't have to be totally cross-section, so it's harder to see. But here you see the axons really nicely cut across. So not many bodies of cells, a lot of the axons, myelinated axons. And the outermost layer of connective tissue will be the epineurium. It will also often have many blood vessels, and it's protecting the whole peripheral nerve. So when you look into the workbook, you see that you should be able to find these 3 layers of connective tissue, which we, I think, did. It's not very complicated. Axons, that's basically everything that's filling the thing, and Schwann cells. So Schwann cells, once again, will be the cells that are wrapping around the axons. So these nuclei will be the bigger ones, will be the nuclei of Schwann cells that are on the section capillary here. So perfect. Peripheral nerve is really straightforward, I think. Then in your workbook you also have a picture from electron microscope, which is similar to what I showed you before. So once again we have a myelin sheath here, second one here. And so this is the sheath, and this is the inside of the axon on cross section. So cut across the axon, we see the membrane and cytoplasm of the Schwann cell wrapping around, and inside you see the microtubules again, and you see one mitochondrion. So it's in the workbook after the peripheral nerve. So yeah, so mitochondrion, cytoplasm of the axon, and the myelin. Myelin sheath here. Now this is the second mitochondrion. It's not really easy to see, but here we go. So nothing very complicated. And now in the final minutes, let's focus on the ganglion. So we talked about the peripheral nerves, efferent together, being carried around the body inside the sheaths of the connective tissue. But I told you that we also have some bodies of neurons inside the periphery. So these will be the ganglions. And oh, one more thing, sorry. So the peripheral nervous system can also be divided, that's what we are getting to, into somatic nervous system and autonomous or autonomic, I'm not really sure, nervous system. So this one is the slow one, the modulating one, sympathicus, parasympathicus. And this one will have ganglia in the periphery. So we will take a look at those. Somatic nervous system is the normal one. The sensory information from the skin, for example, and the efferent motoneurons, like the basic system that's carrying sensory information and by which you are controlling what you do with your body. So the somatic nervous system will be sensory receptors and information coming into the spinal cord, and the information going to the muscles and other effector organs by the efferent nerves. And so in the somatic nervous system, we already saw the motoneurons sitting inside the spinal cord, but there are also cell bodies in a ganglion. We will not be looking at this one. You will be looking at it in the second year histology course. However, you should know that it exists. So it's a dorsal. So we are talking about ventral roots, ventral horns with the motoneurons. Then you have dorsal horn, and you also have dorsal root of the nerves coming out and in, and that's where you have a group of cell bodies outside of the spinal cord as well. And that's where you have cell bodies of the neurons that are carrying the sensory information from the periphery. So, and these are the pseudo-unipolar neurons. I was saying when we talked about the division of the different types of neurons that we have pseudo-unipolar ones and that they can be found in the spinal ganglion. So this is it. This is the place where you would be looking at them. We will not be looking at them today. Instead, we will take a look at a ganglion of the autonomic nervous system. And so this is like sort of modulating system, and it's different because it has two neuronal pathway. So the first neuron of the pathway of sympathicus or parasympathicus is sitting in the spinal cord somewhere, and its axon is coming out of the central nervous system and it's reaching the second neuron, which is sitting in a ganglion in the periphery already. And this neuron, the second one, is the one that will be modulating the peristaltics, the rate of your heart, and function of your salivary glands and stuff like that. So preganglionar and postganglionic neuron, and this one will be in the ganglion, the group of cell bodies in the periphery. We will not get into this. You can take a look, but it's more, I think it's more like physiology or anatomy. So sympathetic and parasympathetic nervous systems I will skip, and we don't need that really now. So we will take a look at one of these autonomic ganglions, and oh, this is in Czech. I'm sorry, I forgot to translate this part, so I will do it later, and I will upload the better version. So autonomic ganglion will be this group of neurons that are sitting in the periphery, and they are reached by axons from the central nervous system, and they produce an axon which is reaching the target tissues, and they are multipolar cells, multipolar neurons sitting in the ganglion. Yeah, so autonomic ganglion slide. Yeah, so this is a Czech lesson as well, I'm sorry. And this would be an autonomic ganglion cut across on a slide, stained with hematoxylin eosin. And what you will have inside are the neurons, which are big multipolar neurons. So you will see the cytoplasm and the nucleus with the nucleolus, and they will be the most prominent cells that you will see in this slide. They are also called ganglion cells because they are big neurons in the ganglion. They are basically making the ganglion, but they are not alone. They also need some glial cells to support them, provide nutrients and stuff like that. And these are called the satellite cells. They really form like a layer around the ganglion cells. So they're like satellites around the Earth or something, so it makes sense. So satellite cells will be the glial cells in the autonomic ganglion, and apart from that, you will also see the non-myelinated axons of the ganglion cells. So it's the autonomic nervous system. Think about modulation of internal organs, so we don't need very fast reactions. We can not spend too much energy and just carry the information more slowly. So non-myelinated axons will be here as well. We will take a look. And inside the cytoplasm, usually you will see something called lipofuscin. So it will be brown, yellowish substance inside the cytoplasm, and this is something that's accumulating in cells that live for a long time. So it's often found in neurons, which live for usually for your whole life, and they metabolize a lot, they degrade a lot, and the residues that come from lysosomal degradation, they are not the cells are not able to use them anymore, they are not able to get rid of them, so they just store it. So this is what lipofuscin is, and it's not functional at all, it's just residue accumulated in the cells. So let's take a look at the slide now. So in the workbook we can see that we should be able to see multipolar neurons, satellite cells, and non-myelinated axons. So the multipolar neurons are the ganglion cells, satellite cells are the glia, and non-myelinated axons will be around. So let's go back to histology guide and let's take a look. What is it? So we don't want we want the sympathetic ganglion, so the autonomic nervous system ganglion. This is the spinal ganglion that you will see in the second here. It's similar anyways. So sympathetic ganglion, and we will zoom in. I'm sorry, I know it's already past the end of the lesson, but we will finish really quickly. So when you zoom in all the way, you see these big round cells with big round nuclei with the nucleolus visible, and inside the cytoplasm you see this brown stuff. So that's the lipofuscin. This is the lipofuscin in the cytoplasm, and these are the big multipolar neurons. So big multipolar neurons, and they, let me find a nice example. They will have the satellite cells around them. Not really nice, but anyway. So yeah, so the glial cells will be forming this like ring around the multipolar neurons. So these cells around the neurons are the satellite cells. So they are really everywhere. Just wanted to find a nicer area, which I didn't really succeed in. Anyways, so you have to be able to find the multipolar neurons. They are everywhere in the ganglion, so you don't have to worry about it very much. You should be able to identify the satellite cells, so the glial cells around the big neurons. You should be able to see the lipofuscin in this slide, and then you should be able to see the axons. So if we zoom out a little bit, you see that parts of the ganglion look like fibers, sort of. It looks like connective tissue, but it will not be connective tissue, it's the non-myelinated axons. So see this part where you don't have the cell bodies? So these are the axons coming from the cells, and they will be going out of the ganglion. So if we zoom in, we will see that it looks like fibrous structure going through the ganglion, and the nuclei that we have here will be nuclei of the Schwann cells, because they are not forming the myelin sheath, but anyway they are there protecting the axons, like we talked about it. This is a blood vessel here, and these are the multipolar neurons with the satellite cells, and these will be the non-myelinated axons. So that's the autonomic nervous system ganglion, and you can find it in here in the histology guide. So in that's it for the workbook, but we also have a mention of the colon here. So that's one more thing I want to show you. So this is more like a repeat sort of thing, because so the autonomic ganglions can also be like smaller and located directly in the wall of the organ that they will be influencing. So in case of the intestine, you have small ganglions that are forming this net inside the wall of the gastrointestinal tract, and it's influencing, for example, the peristaltics. So it's pretty important. So we can just quickly go to the colon slide here. So you have it in histology guide under parasympathetic ganglion. So it will be parasympathetic nervous system, but you don't have to really know that. So in colon, we can open hematoxylin eosin slide, and let's just quickly check where it would be. So you have the mucosa in colon, I'm sure you remember, with all the Lieberkühn scripts. Then you have submucosa, and then you have the muscle. So 2 layers of smooth muscle. And if you want to look for the ganglia of the plexus myentericus, so it's called plexus myentericus, this system of ganglia in the colon and in small intestine as well. So you need to take a look at the place where the 2 layers of smooth muscle meet. So we can zoom in and we start seeing so we start seeing these weird structures that we haven't met before really. So smooth muscle here, and this is not a blood vessel, it's filled with big cells with nuclei. So this is the ganglion of the parasympathetic nervous system sitting in the wall of the colon. It will be influencing the smooth muscle which is directly around it. So this is the ganglion, this is another small ganglion, this is another small ganglion, and they are the second neurons in the pathway of parasympathetic nervous system. They will be producing neurotransmitters around from their axons, and they will be modulating the contractions of the smooth muscles. So it's pretty easy to find between the 2 layers of the smooth muscles, and the cells that you see there are the neuronal cell bodies. So that should be it. So this is the last thing that we carried. You also have it in the presentation, and so you can find the plexus myentericus between the layers of the smooth muscle in the colon. So that's it. So one more check slide for you. So děkuji za pozornost, which means thank you for your attention. So that's it for today. I'm sorry for taking longer. So there's one more question. Okay, what is the function of the satellite cells? It's similar to astrocytes in the central nervous system, so they will be supporting and providing nutrition to the neurons in the ganglion. So the basic function of the glial cell that you would expect. Nothing really very special. So helping the neurons survive and function properly. So that's it. If you have any more questions, you can for sure send me an email, and I hope we got to everything we needed to today. It's not optimal with the slides. I will try to get the virtual slides that we have at the faculty so that I would be able to show these to you as well from my home, but so far I don't have them. So thank you for watching, and so next yes, one more thing. Somebody asked me via email if this video will be available for download. Yes, it will. I also recorded this, so I will upload it to Vyuka. So you will be able, if you didn't catch me, if you overslept, understandably, you will be able to see it later on. And so therefore, if you have questions, then just email me. Next week it will be Mrs. Hubičková who will be doing the practice for non-specific immunity, and I think she will just be recording it once and uploading it. But she should I think let you know how this will go. And and the week after that, for specific immunity, it should be me again. So I will let you know in advance about the streaming, but I think I will do it the same way as I did today. So thank you very much, and take care and stay quarantined, and bye.